human tumor tissues across 17 different cancer types. Our research has helped many biotech companies in progressing their pipeline candidates to and through clinic. We are focused on creating a platform that allows us to preserve and modulate the human tumor microenvironment with a variety of therapeutic regimens. I'm happy to introduce today Dr. Nandini Pal Basak, Director of Farcast R&D, who will present our latest research that was accepted as a poster at the recently concluded AACR meeting last week. She will present data on Farcast's proprietary human histoculture system and demonstrate the functional fidelity of tumor-infiltrated leukocytes during a 72-hour culture period. Nandini has been part of the Farcast team since 2017 and is an expert in the field of cancer biology. Her PhD thesis focused on cancer metabolism, and she has hands-on experience in a variety of assays such as flow cytometry, nanostring, cytokine imaging, and immunohistochemistry. At the end of this presentation, we will have a 10-minute Q&A sessions. So please feel free to post your questions in the chat box or email these to biopharma@farcastbio.com. Please welcome Nandini. Thank you.
Thank you. Hello. I hope everyone is safe in these unprecedented time of global pandemic. Like last year, this year also we had the AACR in virtual mode, and I'm sure some of our audience have attended it and had a chance to visit our poster. So in the second version of our webinar, what we want to do is walk you through our poster, and we would love to interact and have some questions from you. So this is our poster titled Understanding the Functional Fidelity of Tumor-Infiltrated Leukocytes in a Human Tumor Histoculture Platform. Farcast Time. So Farcast Time is a histoculture platform, and this is a brief understanding of our workflow. So we get tumor samples along with matched patient blood from various hospitals, and once it comes to our facility, we process it such that we make small fragments. These fragments have the tumor microenvironment preserved. Then we can culture it. Over a short period of time, and during this culture time, we can treat it with various drugs and its combination. Once it is treated over the 72-hour culture period, we can interrogate it with various assays and try and understand how the treatment has affected the mechanism of action and the various immune compartment or the tumor in this tumor microenvironment. In this study, we have focused on head and neck cancer, and here in the top panel, what you see is a head and neck sample, H&E, where pre-culture you see the tumor, the stroma, and the various immune compartments in it. Post-culture, we are able to preserve this architecture where you see the tumor along with the stroma and the immune cell in it. So this immune compartment is very well conserved, as you can see by the R-square value. And though there might be some loss in the immune compartment, it happens in a uniform manner, and there is not any particular immune compartment which gets affected any more or less. Now, this immune compartment we observed is live. So this is a flow cytometry data at 72 hours of after 72 hours of culture, and we can see live T cells, various compartments of T cells, then the myeloid macrophages, NK cells, and monocyte and neutrophils. So these live immune cells are present in our culture after 72 hours. The question we ask is, are these functional? So to understand it, we treat it with various stimulants like anti-CD3 plus IL-2 to stimulate the T cell compartment, or LPS to stimulate the macrophage compartment. Post-treatment, we then try to understand this using various cytokine analysis, flow cytometry, and nanostring analysis. So first, with the cytokine data, what we observed that upon these treatment, there is a very specific response. That is, with anti-CD3 plus IL-2 treatment, we found specifically the T cell cytokines are upregulated. As you can see for granzyme B, perforin, and IFN gamma, which is not responding when it is treated with LPS. On the other hand, when it is these explants are treated with LPS, we find a very specific response of cytokines related to macrophages like TNF alpha, IL-6, IL-10, and IL-8. Thus, it shows that in our platform, these stimulants are able to stimulate the immune cells, and also it does in a specific manner. This is further observed in the flow cytometry analysis, where you can see activation markers like proliferating CD8 cells or activation marker expression of CD69 or intracellular granzyme B increases only upon anti-CD3 plus IL-2 treatment and is not affected by LPS treatment. Even by nanostring analysis using various gene signature, we observe this phenotype where you can see there is an increase of T effector by Treg ratio upon CD3 plus IL-2 treatment, whereas the IL-1 signature, that is IL-1 alpha and beta, increases upon LPS treatment, which is not that much affected by anti-CD3 plus IL-2. This shows that these immune cells and the tumor are preserved in our culture. The immune cells are viable and functional. Next, we ask, if we treat it with an immuno-oncology drug, will this immune cell respond to it? So to understand that, we treated these explants with nivolumab, which is an anti-PD-1 therapy. Now first, for each and every sample, what we try to understand is whether this nivolumab is going inside the tumor explant and binding to its target, that is PD-1. So in this representative graph in flow cytometry, what you observe is you have the CD3 plus PD-1 plus population, which is masked upon nivolumab treatment, which shows that nivolumab is entering the explants and binding to its target. This is observed for all the samples. But the CD8 reinvigoration phenotype upon nivolumab treatment is not observed in an uniform manner, which we monitor by IFN gamma secretion in these samples. So here in this waterfall graph, we have plotted the IFN gamma secretion over this 72-hour culture, the log to fold change with respect to control. And what you observe, there are some samples where there is a high amount of IFN gamma secretion, whereas there are some samples where there is negligible change in IFN gamma. So for our further understanding of nivolumab response, we designate sample 41 as a responder and sample 38 as a non-responder. And if you now see in this kinetic representation, how the IFN gamma behaves in these two samples. So the bottom panel is the responder panel, and that is sample 41, and the top panel is for the non-responding sample, that is sample 38. So you observe clearly that there is a very nice increase and steady increase of IFN gamma in sample 41, which is not observed in 38. Similarly, for granzyme B, we observe that with time, granzyme B increases more than the control sample, which is not observed in 38. And also for perforin, we observe a similar phenotype. For the with nanostring also, we try to monitor whether we are able to capture this CD8 reinvigoration using various gene signature. As you observe for IFN gamma or tumor inflammation score or the CTL signature, there is an increase in sample 41, whereas such an increase is not observed in sample 38. We also focused on these gene signatures like apoptosis or cell proliferation, and we observed in sample 41 apoptosis gene signature increases, but in sample 38 it does not. So we wanted to explore this further. So in this particular graph, so we wanted to understand whether this apoptosis is happening in a tumor, is it happening in the tumor compartment? To understand that, we monitored the tumoricidal effect by doing cleaved caspase tree IHC, and our in-house pathologist scored it in the tumor compartment. Now, what you observe in sample 38 is that there is hardly any change in the cleaved caspase tree expression pattern upon nivolumab treatment. Whereas in sample 41, we see an increase in the mean of cleaved caspase tree expression in comparison to the control. This clearly shows that during the short time duration of nivolumab treatment, not only the early phenotypes like cytokine increases and gene signature changes is observed, but also we start observing the tumoricidal effect. Now, with nivolumab treatment, another phenotype is commonly reported, that is immune cell infiltration in the tumor nest. To understand this particular phenotype in our platform, we did multiplex IHC from FFP block samples. So in this particular image, what you observe is CD8, which is marked in green, pan-CK, which is marked in red, and so if you see the control panel, and there is hardly any immune cells or CD8 cells within the tumor nest. But upon nivolumab treatment, for the responding sample, that is 41, the CD8 cells have now infiltrated within the tumor nest. Whereas this type of phenomena is not observed in the non-responding sample. And we also did a quantitative evaluation of this particular phenotype, and we observe a significant increase in this infiltration of CD8 population in the tumor nest. Thus, all these observations are pointing towards the fact that in our platform, we are able to see the mechanism of action of nivolumab, and as we know, these anti-PD-1 therapy may not respond in all the samples. And thus we see a variation in this response pattern with respect to the CD8 reinvigoration. We next wanted to understand whether the baseline gene signature had some differences in between these two samples. So if you look at sample 38 and 41 immune cell scores as determined by nanostring based analysis, we observed that sample 41 was a much more immune hot sample compared to sample 38. And we also see in the baseline that it had a higher IFN gamma signature. The tumor inflammation score was higher. Even as we know for various drug treatment, hypoxia is kind of a resistant mechanism, and we observed that in sample 38 there is a higher hypoxia signature compared to 41. For PDL1, PDL2 signature, that also correlated very well. That is, sample 41 had a higher PDL1, PDL2 signature compared to 38. Now, the classical way to determine or the way anti-PD-1 therapy is provided to patient is by observing the PDL1 IHC at baseline. And as you observe, for these two samples, the pathology scoring, or that is the CPS scoring, was actually comparable in the two samples. Thus, from this study, we understand that just by observing PD-L1 expression at baseline by IHC may not be sufficient. A more multidimensional way of understanding how the microenvironment gets altered by these anti-PD-1 therapy may be more insightful, which we can observe from our platform. Next, I would like to share a case study on a combination therapy, that is nivolumab and ipilimumab, which is currently, there are many clinical trials ongoing, especially in head and neck samples. So in this case study, I present one sample data where you can observe that with nivolumab or nivo plus ipli, there is masking of the PD-1, and also, as we know, ipilimumab has an effect on depleting the Treg population. And here in this particular sample, what we observe that the Treg CTLA4 positive compartment is depleted upon nivo plus ipli combination treatment, which does not happen upon only nivolumab treatment. Next, we wanted to understand what is happening to the various immune compartments. Is there a signature which is defining how nivo plus ipli is working? So what we observe that upon nivo plus ipli treatment, there is a decrease in the proliferating Treg population, as you see in this blue graph. And there is a comparative increase of the proliferating CD8 more than the nivolumab treatment. So thus there is a tilt towards more proliferating cytotoxic T cell compared to the regulatory cells. Again, we observed that upon nivolumab treatment, there was an increase in the CTLA4 expression, as you observe over here. But upon nivo plus ipli treatment, the CTLA4 expressing CD3 compartment is definitely depleted or reduced. But on the other hand, we start observing another checkpoint molecule, that is TIM3 expression going up. So this kind of tells us what is happening in the microenvironment upon treatment with these IO therapies, either as monotherapy or in combination. Lastly, we wanted to understand whether nivo plus ipli is helping in better CD8 reinvigoration. So as you observe for this particular sample, nivolumab had hardly any impact on these CD8 reinvigoration, whereas upon nivo plus ipli treatment, we start observing a better response on IFN gamma secretion. Similarly, we observe a better response of nivo plus ipli with respect to perforin. We hopefully will have more data with this combination, comparing the combination of nivo plus ipli, and maybe soon we can present that data to you in our subsequent webinars. Thus, to conclude, the Farcast time histoculture platform is able to preserve the tumor morphology and the various immune compartments and retain their functionality during this culture period. In this near-native microenvironment, one can explore any novel immuno-oncology-based therapies and compare it with existing standard of care immuno-oncology agents, like we showed over here, nivolumab, which is a FDA-approved therapy for head and neck cancer. And also with these interesting insights, we understand that this particular platform has the potential to predict the response to immune checkpoint inhibitors, and that is something which will be one of our focus in the near future. I would be happy to take some questions. Okay, so we have a question. What are the various other mechanism of action that are being tested in this platform? So apart from immuno-oncology based therapies, we are also testing targeted therapy. Like one of our R&D work is going on to understand how we can measure cetuximab response, which works by ADCC mechanism. And also we in the past webinar we have shown how we have worked with chemotherapy, which specifically we are observing tumoricidal effect and effect on like the viability of various immune population.
Nandini, while this research was done on head and neck cancer, uh, does this platform offer other cancer types as well?
Well, very good question, because we started with head and neck for reasons, because head and neck is a kind of tumor in which there is a lot of immune population. There has been studies which has been published in various journals talking about the immune populations which are present in head and neck. And so we started with it because it kind of provided a very good model system where there are a lot of therapies with different mechanism of actions approved. But nonetheless, we are currently optimizing various other indications like CABR or breast cancer, gastric cancer, especially stomach adenocarcinoma, then renal cell carcinoma, and also ovarian cancer. So these are some of the platforms which we are currently working on, and hopefully soon we'll be able to launch these different indications. Okay. So can this platform be used to study immune cell homing? Okay, so that's a very, very interesting question, because the way we have approached this platform is one, to keep the inherent immune cells intact. So the study which I presented here, all the phenotypes which we are observing is due to the inherent immune population. Now, various other mechanisms could require immune cells from the peripheral blood to be homed in, or there will be signals for it to infiltrate within the tumor. So for doing that kind of study, we have the option of adding PBMCs or with nucleated cells or mononuclear cells. And we can study this, and the way we are developing the assay, we have you can stain these PBMC with any dyes like CFSC and monitor its homing, and compare with the separate out the TILs and see how these immune cells which are coming in from the periphery affecting the tumor microenvironment.
Nandini, would it be fair to say that this platform, when compared with other modes such as cell lines or xenograft models, fares better? And if you could explain that a little bit.
I wouldn't say better or worse or anything of that sort, because each and every model has its own importance in the journey of, for example, a drug development. So each model has its advantages. Like when we talk about cell lines, it gives you high throughput and gives you preliminary data, but it will not give you the complexity which is present in an actual patient tumor microenvironment. Now, when we come to this histoculture platform like Farcast time, these maybe you cannot do a highly high throughput study, but you can be able to understand what is happening in the patient tumor microenvironment, because we are able to preserve the nuances of these microenvironment in our platform, and therefore you will understand that when you are giving a drug, this complex microenvironment, how is it affecting the functionality or the effectiveness of the drug. So so that way, so it depends on what question you are asking. So our platform becomes much more important when you are translating from during the translational research period. I would like to put it that way. Okay. Okay. The next question, are you able to observe modulation in all sorts of immune cell population, myeloid and lymphoid? Yeah, so that's again a very important question, as we showed that these immune cells which are present using known stimulants, we are able to stimulate and modify it. And through these checkpoint inhibitors, we have demonstrated how the T cells are getting modified. Now we are also conducting a study where we are trying to modulate these myeloid compartments to do this polarize the macrophages. For example, if we can increase kind of move from M2 to M1 and see the impact it will have on the tumor microenvironment. Is so okay, is there data to show reproducibility of response to drug treatment? Okay, so so during when we were standardizing our platform, the way we approached this was to do these drug treatments in duplicates and try to see that the response which we are getting in one arm, is it happening in another arm? So in that way, we have tried to observe this, and yes, we do have some data to show that this response is reproducible across in a particular sample. Yeah, I think we are already running almost, we have come to the end. So I would like to thank you all once again for attending this second episode of our webinar series. And if you have any other queries or any other questions about our platform, please do write to us at biopharma@farcastbio.com. Thank you.